Thank you for joining us today. We're delighted that Dr. Derek Leishman, Vice President of Translational and Quantitative Toxicology at Lilly, will talk to us about the opportunities to leverage non-clinical data in a regulatory integrated QTc assessment. Also, Steve Jenkinson, Vice President of Drug Discovery and Safety from Metrion Biosciences, will discuss a clinically translatable human iPSC cardiomyocyte model to predict QTc and QRS cardiac risk. So if you have any questions for the presenters, please enter it into the questions pane, and we'll answer as many questions as we have time for. Okay. Before I invite Dr. Leishman to give his presentation, I'll just give a very brief overview of Metrion Biosciences. So we're a specialist preclinical CRO. So we have expertise in ion channel screening, neuroscience assays, and relevant to this webinar, cardiac safety screening, including GLP-HERG, SIPA panel, and phenotypic cardiac assays using human iPSC cardiomyocytes. Okay, so our first presentation today will be Dr. Derek Leishman. So I'll just firstly introduce him. So Dr. Leishman gained his experience as pharma topic leader and past rapporteur of the ICH E14 S7B implementation working group. And in that capacity, he'll share the opportunities presented by the new ICH E14 S7B Q&As. Dr. Leishman is also past president of the Safety Pharmacology Society and has spent 32 years in the pharmaceutical industry, primarily focusing on cardiovascular safety and PK/PD modeling.
So really what I'd like to do today is to just talk about some of the opportunities that have been created by the new Q&As, and it's really an opportunity to improve the efficiency and the efficacy of QTc assessment, starting with the very earliest in vitro studies. This is just a disclaimer, as Eddy mentioned. What I'm really talking to you about is not really in my capacity as some Lilly scientist, but more in my experience as the past rapporteur and a member of the ICH E14 S7B group, as well as somebody who's applied these principles and done these QTc assessments for the last 25 years. So speaking of 25 years, if we look at the regulatory timeline, this is a topic that stretches back all the way back to 1996, where the European Medicines Agency issued a points to consider document that was followed by white papers from both FDA and Health Canada. And then this is a topic that stretched from 1996 to the current day. And you can see along the way there has been some changes and an introduction to an ICH process that started with ICH S7A. So in 2005, ICH E14, not 15, E14, and ICH S7B, those were really the first elements that introduced what I call the TQT era. So a TQT was required regardless of S7B results. So everybody was doing these large, expensive clinical pharmacology trials in addition to the in vitro HERG assay and an in vivo QTc assay. There were some changes to a Q&A for E14 Q5.1, and that ushered in what we'll call the sort of end of the TQT era, with concentration QTc being an acceptable endpoint in a clinical evaluation. That also allowed the use of early clinical studies to be used instead of a dedicated standalone thorough QT study with moxifloxacin as a positive control. Then in 2022, there were some Q&As introduced. The Q5.1 was updated for E14 along with Q6.1, and then for the very first time, ICH S7B had some Q&As introduced, and that really started what we're calling now the integrated assessment era. And that's where your non-clinical data can really start to have an impact. So now, rather than a clinical evaluation being conducted regardless of the S7B results. The clinical data can be taken along with the clinical results to support your QTc assessment. And so, as I mentioned, that's the 2015 change with concentration QTc really ushered in the end of the TQT era. So here's some data that was presented by Christine Garnett from FDA, and it's looking at the different types of QTc evaluation that the FDA integrated or independent review team had looked at over the course of 5 years, actually 6 years from 2016. So they starting with the next year after the introduction of the concentration QTc. And what you can see here is that the different types of assessment, TQT is the red bar, and then assessments under Q5.1 and Q6.1 and E14, over the course of these 6 years, what you can see is that the TQT numbers were high initially, 62%, whilst 5.1 were very low, that really hadn't taken any effect then, to now. And now what you can see is that we're in a situation now where the assessments conducted under Q5.1 and the concentration QTc has now, for the first time, become more abundant than the TQT assessments, and that's a trend that we expect to continue. What's interesting in this slide too is that under Q6.1, and I'll talk in a minute about what Q6.1 is, the number of assessments that have been done under Q6.1s are relatively constant, 25 to 30%. So this is just a slide that kind of summarizes some of the options you have, but also options that have changed with time. So the options that we have always had under ICH E14 are, what I'm calling the status quo. So you can do a standalone moxifloxacin controlled thorough QT study and make that your QTc assessment that the FDA and others will use to determine how many ECGs you need in your phase 3 studies, and ultimately what your label looks like for your approved product. It's also the case that in certain situations you just can't do a thorough QT study. So that's perhaps in a situation like oncology, where you can't do a placebo-controlled study because the toxicity of the agent is just too high to be really considered for a placebo group, and also it's unlikely to be done in healthy volunteers. So those are situations that make it a little difficult to do a moxifloxacin controlled thorough QT study. So those are covered by ICH E14 Q6.1. So those are the cases where a conventional QTc study isn't feasible, and that's the group of assessments that have been a kind of constant 25 to 30%. The other 70% were really being dealt with under the first option, which was the thorough QT study. The end of 2015 ushered in the update to Q5.1, the use of concentration response modeling, and that now allows concentration QTc assessment from the early clinical studies to be used as your clinical assessment. So you're going to do those early studies anyway by doing slightly different ECG recording and doing concentration QTc analysis of that data. That first in human study that you were going to do already can stand in place of the thorough QT study. However, just because it's not supported with a moxifloxacin positive control, there is some stipulations on the concentrations you have to achieve, and essentially you have to achieve 2 times the highest clinical exposure you're going to try and support in your QTc assessment. So that was the one additional criteria that was applied in the concentration QTc modeling. What happened in 2022 with the integrated risk assessment Q&As, you now have the opportunity to support Q5.1 with the non-clinical data, with a hERG negative and an in vivo study negative, and you do that in situations where the tested exposure in the clinic doesn't achieve the 2 times the high clinical exposure. So if you find yourself in a situation where you haven't covered 2x the high clinical exposure with the concentration QTc modeling, then you rely on your non-clinical data and a negative in the hERG study and a negative in vivo study to support your QTc assessment. It's also the case that if you were using the Q6.1, where you couldn't do a conventional QTc study, the normal label that appears for the product under those circumstances is that the QT is unlikely to be larger than 20 milliseconds, assuming that you saw nothing in the QT assessments that you did manage to do under Q6.1. If you saw nothing, then the conclusion would be that there was unlikely to be a QTc change bigger than 20 milliseconds. That can actually be updated to a label suggesting that there's a low risk of any QTc prolongation, provided your hERG study is negative and your in vivo study is negative, and you can demonstrate that your in vivo study is sensitive and rules out QTc effect at the same kind of sensitivity as the clinical study. This is all still very much anchored on QT. I know these are pro-arrhythmia assessments, and there's a lot of discussion about QT being a relatively poor biomarker for proarrhythmia, but QT is the hazard identification, if you like, and once you've crossed the line and you're in the territory of a hazard, then you have to start doing a more active proarrhythmia assessment. But it's the case that most compounds don't have that hazard and don't prolong the QTc interval, so it's really that first step in the assessment. So it's all anchored on QT still. The ICH S7B Q&As, they provide some information on how you do the best practice for the hERG assessment and the in vivo study. So if you're going to support those, use those to support your clinical assessment, you have to make sure those are conducted to best practice, and that's really providing the best quality numerator for a margin calculation. The denominator comes from the clinical exposures. Clinical concentration QTc assessments has provided an understanding of the concentration QTc relationship for some reference agents, and we now know with some confidence what the concentration associated with 10 milliseconds QTc change in the clinic is for 3 reference agents, dofetilide, moxifloxacin, and ondansetron. And this is a clinical benchmark which you could verify again. You could run another moxifloxacin study and see if you could confirm these concentrations. So it's a very easy to support benchmark to be used, and so that's the benchmark that's used for reference agents in terms of exposure. For the test agent, you would use the high clinical exposure as the margin denominator. So the best practice is providing you with the numerator for margin calculations, and the denominator is the high clinical exposure for the test article. The nice thing relative to hERG in particular for those 3 reference agents, there were 3 agents where there was good clinical information to find that benchmark at 10 milliseconds QTc change, but they also happen to have different kinetics of hERG block. So they kind of spread across the spectrum of hERG properties, from compounds with slow kinetics like dofetilide to those with fast kinetics like ondansetron. So one of the things that is immediately noticeable with the ICH Q&As and the training materials that go alongside it is that we're no longer dealing with margins as a point estimate. Everybody's been very used to taking their hERG IC50 and dividing it by the mean Cmax unbound concentration of a molecule, and so you get a point estimate of the hERG margin. But that point estimate doesn't take into account some of the variability that's inherent in those input data. So that has been changed in the new Q&As. So now you use the HERG IC50 along with the distribution or the standard deviation around that to create a distribution of HERG IC50 numbers, and those are shown on the left hand side in the top. You can see a simulated distribution based on a mean and standard deviation, and it's 10,000 points. And then below it is the drug concentration. Again, it's the mean Cmax with the standard deviations, and you can create a distribution of those. And the same for the fraction unbound in a plasma protein binding experiment. You can figure out what the mean is and the distribution around the mean. And you could sample from those three different distributions, and you can come up with the HERG margin distribution on the right. So the mean margin in this case is around 20, but you could see it could vary anywhere from 10 to nearly 40. And this is a margin actually based on the data for moxifloxacin that's in the training materials. And what you would be looking for in terms of determining whether or not you had a HERG negative is the lower bound of your test article margin distribution versus the upper bound of the reference compounds distribution. So you really look for those two margin distributions to not overlap, and that would give you your HERG negative. So there are some best practice considerations for the HERG assay given in the Q&As. There are considerations around temperature, the voltage protocol used, and then concentration verification, making sure that the compound was actually what you think you used and tested in the assay is what was actually tested. There's also some information to address the reporting so that the quality of your study is more transparent. And then one of the other changes that's different from previous iterations of the HERG assay is the use of two concentrations of a positive control, one at a high concentration, one at a lower concentration, so you really bracket the IC50 for your positive control, and that just allows a better assessment of the relative potency of the positive control. And of course, the comparison that you're going to be making is to the reference compounds, which will have been recorded using the same protocol in the same laboratory. There is the ability to borrow data from different laboratories as long as the same protocol is used and there's at least one common reference agent used. A sponsor, of course, is also perfectly at liberty to use any protocol they like rather than the best practice protocol, but then the obligation on the sponsor is to build sufficient reference compound information to show how the reference agents would have responded under that protocol and to build the reference compound margin that way too. In vivo assessment's not really the topic of today's talk, but I've included this picture just to illustrate that there's been a migration in how we're thinking about in vivo margin. Anyone who's followed the ICH E14 knows it's very strong on the 10 millisecond threshold in the clinic. There's a recognition in the animal studies when you compare those to the clinic that really the sensitivity to detect is modulated with the multiple of the clinical exposure that you test. If you can test a very high multiple of the clinical exposure, then you don't need an assay that's quite as sensitive. If you can only test a modest multiple of the clinical exposure, then you have to have really quite a sensitive in vivo assay. And that's what's shown in the picture on the far right. What you really want to be is to the right-hand side of that diagonal line, so that if let's just say your minimum detectable difference in your study was 15 milliseconds, you'd really want to be able to test somewhere in the region of 5 to 10-fold the clinical exposure to give yourself some reassurance that your in vivo negative really was a negative. And these concepts are discussed in the reference at the bottom of the slide. There are some emergent gaps, and I think some people on the call today may find themselves dealing with some of these gaps, because you really end up in the Q5.1 and Q6.1 scenario where you have limited clinical exposure, and you usually have limited clinical exposure when you have some tolerability issues. Now, those tolerability issues in man could equally apply to an in vivo animal study, and so it may well be that you find that you end up with an in vivo study that can't test high concentrations. So then what do you do? And then there's there's the situation where you've got the in vivo study, it has either a poor detectable threshold or some limited exposure, or actually heart rate changes which can confound the heart rate correction of QTc, and so you end up with some degree of uncertainty based on your in vivo data. So it's not uncommon to bump up against these gaps. So how do you bridge those gaps? Well, the ICH discussions and the regulators present and the industry representative groups, nobody was really interested in repeating an in vivo study. That just seems to be going in the wrong direction from a 3Rs perspective. But we do acknowledge that there are in vitro human cell assays and human tissue assays which are available. For example, a cardiomyocyte with an action potential. So that's a human-based integrated assay that could serve some purpose in helping bridge the gap when the in vivo study can't be conducted for the reasons I alluded to in the previous slide. There are principles in the ICH-S7B Q&As, so those are question 2.2 and onwards, and question 4, which kind of cover some of the principles that you'd have to apply to using a novel assay to support the QTc assessment across the gap, and the ability to detect the effect at the critical concentration for the 3 reference agents is obviously key. Those are the reference agents that we know prolong QTc in the clinic, and we know at which concentration they prolong QTc in the clinic. So understanding how they behave in any novel in vitro assay is critical to making some sort of assessment. So this is my last slide where I'll be speaking, and it's really just to wrap up with the conclusion that the new Q&As for ICH-E14 and S7B really do offer the opportunity to be more efficient, because you can use concentration QTc, and you can use your in vitro and in vivo data to support the QTc assessment. All those studies were happening anyway. Now, with a little bit of work in terms of best practice, you can make them count against your QTc assessment that everybody's doing at the end of phase 2. We've got the margins for the key reference agents, so we know what those look like, and it's possible to then define what a negative looks like in vitro and in vivo based on best practice assays and based on a multiple of clinical exposure. A positive finding in either the HERG or the in vivo, of course, can still be negative as long as the margin's big enough. That's one of the things that's kind of interesting in this. You can have a HERG IC50, but with a big margin, it's regarded as negative. You could have a QTc effect in an in vivo study, but if it's at a large clinical multiple, then it's also regarded as negative. So even positives can be negatives. And then if necessary, because of tolerability or heart rate effects, you can use an in vitro integrative assay and use that in place of those in vivo studies or alongside those in vivo studies to help with the overall QTc assessment. And this slide just has some extra information. I believe these slides will be available to everybody after the webinar. This has some extra references alongside some ICH materials, and here is an illustration of some of the links that are provided by FDA on the FDA website, where you can go and find the extra information. You can also find templates that you have to fill in for these integrated risk assessments. So you can use those to complete those. And if you download those, you could obviously use those as a bit of a guide so that you can figure out what information you need and what information you have. I will say that in having tried many of these integrated risk assessments recently, probably the piece of information that comes latest for most teams is what the high clinical exposure actually is. So that's the piece that you'll be waiting for quite some time. So you can obviously work out margins and exposures based on the clinical data you have, but getting to that definitive high clinical exposure is probably the biggest challenge I've seen for teams thus far. And so with that, I'll leave the presentation now and hand over to Steve.
Okay, so yeah, so now we'll turn to Steve Jenkinson. So Steve has a strong background in drug discovery and secondary pharmacology, and he's managed drug discovery programs across multiple therapeutic areas and stages, ranging from target identification to nomination of candidates for preclinical development. And he has over 25 years leading discovery projects within GSK and Tanabe research laboratories. And Steve joined Metrion from Pfizer, where he was senior director, and he established, developed, and led the in vitro secondary pharmacology team. Additionally, Steve was co-chair of the IQ DrewSafe secondary pharmacology working group and was a member of FDA's HESI pro-arrhythmia group.
Okay, well, first of all, thank you, Derek. I think that was a great overview of some of the changes that have been happening within the ICH guidelines. And, I think towards the end of your presentation there, you kind of like alluded to, the utility potentially of cardiac, using human iPSC cardiomyocytes as a potential model as well to try and address looking at QT and QRS, in this case QRS risk as well, and that's really where the presentation I'll be covering today is focused on. So just to give you a little bit of background, when it comes to looking at human iPSC cardiomyocytes and defining cardiac risk, it's really kind of routine now across the industry with a number of models, and most large pharma will have their own flavor of this that they've developed over the years. The models are really useful in a couple of aspects. First of all, looking at functional endpoints. So you can look at contractility. Even though the cells are immature, there are methodologies to mature the cells to look at that as a potential endpoint and understand the effects of compounds on that functional aspect. But the focus today of this presentation is really going to be looking at the electrophysiological endpoints, and again, focusing on QTc prediction and QRS risk assessment. Some of the changes that have been happening over the years are that the regulatory body, as Derek pointed out, are really starting to look more at the in vitro aspects of the analysis preclinically to drive decision making. And this, when it comes to stem cell cardiomyocytes and specifically looking at QT liability, a lot of this work was driven initially by the CIPA initiative. And it's been more formalized more recently by the inclusion of a whole section in the ICH-S7B Q&As that's focused on stem cell cardiomyocytes and their utility in assessing QT risk. And the aim here, again, similar to the other the hERG profiling as well as in vivo studies, is to really be able to use this data to help support the thorough QT waiver application, so you can get away from having to do an unnecessary TQT study. Now, one of the things that's very clear in the ICH guidelines, and really is just basically good science, is a key requirement of any of stem cell cardiomyocyte assay to have a really clear understanding of its translation to the clinic. It's one thing to be able to generate data, but if you can't really understand the translation of that data to clinical setting, then it really doesn't have much value. So today I'll be presenting on a high throughput stem cell cardiomyocyte assay that, first of all, can predict the exposures of a novel compound that would be associated with that 10 millisecond change in the clinical QTc interval that the FDA really is focused on. Also define the probability of QRS prolongation risk. And there's a slight distinction there between prediction and probability, and I'll talk about that a little bit later. Also, this assay is generally very useful at just assessing cardiovascular toxicity liability, so less of the safety pharmacology aspects and more of the toxicity aspects. What's nice about the assay is you can use it acutely, but also you can look at chronic endpoints. 24 hours is where most of our translational work has been done, but you can extend it out to 72 hours or longer to look at longer term effects if you are more interested in potential tox liabilities. And what's nice about the assay is it's performed in serum-free conditions, so you really can understand, the free concentration of the compound. And one of the aspects of the ICH guidelines is that you really should be measuring the compound concentration in your assay, so you can really determine the accurate concentrations that the cells are exposed to. And that's always an option with this assay. It's not something you would do routinely because of cost and time, but it's certainly something that this assay is very amenable to, and we have done that in the past as well. So just a little bit more about the assay. So what cell system are we using? So as with most companies at the moment, we're using the iCell square cardiomyocytes from Fujifilm CDI. We've done some analysis on these cells historically and published on that, and most of the data that I'll be sharing today is from this paper that we published earlier, Kilfoyle et al. Back in 2021. And when we looked at the stem cell cardiomyocytes, we compared these to purified human ventricular cardiomyocytes that we obtained from Anabios. And what we found was that when we looked at the gene expression, the key ion channels that are involved in action potential generation had a very similar expression level. So NAF 1.5, CAF 1.2, and the HERG channel all showed extremely similar expression levels. And for the channels where you'd expect there to be a difference from looking at historical data, we observed that as well. So for example, the KCNJ2 that is responsible for the IK1 current, the expression is higher in the human primary ventricular cardiomyocytes versus the stem cell cardiomyocytes. And this, is being well noted historically and is responsible for the higher resting membrane potential that's observed in the stem cell cardiomyocytes. But in general, the HERG, NAV, and CAV are expressed at very similar levels as you'd expect as you see in the human ventricular cardiomyocytes. Looking at the technology that we're using for our assessment, we're looking at action potential recordings, and it's from intrinsically paced human stem cell cardiomyocytes. So the cells are pacing themselves. There's no extrinsic pacing. And we're using the Volta fast optical reader from Lumencore, and that's just showing here on the right-hand side, in combination with using a voltage-sensitive membrane dye, also from Lumencore, and this is the burst dye. What's nice about this instrumentation is it allows us to simultaneously read 96 wells of a 96 well play at very high frequencies, so at 10,000 hertz. And the advantage of running at 10,000 hertz is it really gives you recordings that are equivalent to in quality to patch clamp, and that can be seen from the traces below here from the instrumentation. And you can see that very nice action potential waveforms with a compound that produces an action potential prolongation. You see a nice prolongation of the action potential, which ultimately, when you get to higher concentrations, you'll start to pick up these early after depolarizations as well. So what endpoints are we looking at? Well, we normally perform 10-point concentration response curves for our compounds, and in the next couple of slides I'll explain why we do such a definitive concentration response curve analysis. But the endpoints we're measuring are action potential duration, and we can measure multiple action endpoint durations. The standard one we utilize is the APD90, but we can use the other action potentials to durations in order to determine triangulation if that's an endpoint that the client is interested in. We also look at rise time, which is the time from the initiation of the action potential to the peak here. It's just the time across the bottom here, and also beat rate as well. And again, just to reiterate, we look at this both at 30 minutes and 24 hours for the same cells with the same treatment. And again, the assay is protein and serum free. So just give some example data here. So, there's some different classes of compounds that produce hERG inhibition. So obviously a standard one here is dofetilide. As expected, you see a nice concentration-dependent increase in action potential duration, with ultimately some early active depolarizations occurring at the higher concentration. For calcium channel block with Cav 1.2, if you're looking at nifedipine, you see a nice action potential shortening as expected. And then for compounds that can prevent hERG trafficking to the membrane, ultimately what happens over time is you end up with recycling of the hERG channel, but no replacement. So you end up with an ultimate, basically a reduction in the amount of hERG channel on the surface. So it's equivalent to a hERG block in some ways. And if you look at acutely with a compound such as pentamidine, you don't see any effect on action potential. But the data I'm showing here is after 24 hours. And so with pentamidine, you see this large prolongation in action potential duration as you prevent hERG trafficking to the surface. So the pharmacology kind of makes sense with just some of the basic compounds. What's also nice about this assay is you can actually do a rate correction, similar to as you would do in the clinic or in vivo study. So for example, on the left-hand side here, we're looking at the uncorrected data. So if you're looking at APD90 on the y-axis, and on the x-axis, just beat interval, so R to R interval. And you can see as a beat interval increases, as expected, APD90 increases as well. You can use a standard Fredericia correction. It does a reasonable job. However, if you actually use the Yamamoto correction, which is more designed for stem cells, you end up with a much more robust correction of the APD90. And we actually use this Yamamoto corrected APD90 as a surrogate of our clinical QTc interval. So that kind of summarizes a little bit of the assay and the background behind it. So how do we use this to assess compounds? Well, firstly, we use it to assess clinical QTc risk. And this slide is a little bit busy, but I'll take you through it, and it kind of summarizes how we use the data. So first of all, the validation of the assay was performed using 23 QTc positive reference compounds. And those compounds had, which is important, had available clinical data. And these are the reference compounds here on the right-hand side. A number of them are part of the CYP28 compound list. Not all of them, because not all of the CYP28 compounds had clinical data that we had available to us. So using these 23 compounds, we performed concentration response curves in our stem cell assay. And this is where we use the 10-point concentration response curve with the compound across the bottom. And we're looking at changes in the Yamamoto corrected APD90 here in this case. And this could be also rise time or other endpoints, but in this case, it's just the Yamamoto corrected APD90. We don't look at EC50 values because you don't always get a nice dose-dependent curve. You will sometimes see bell-shaped curves. So what we used as a measure is a concentration associated with the stem cell APD90 threshold value. And this was an arbitrary value we selected, which was 3 times the vehicle standard deviation of all our historical data. And so we use this value here where it crosses this threshold and calculate that value. And we compare that to the free clinical exposure producing a 10 millisecond change in QT from the clinic. So the stem cell data is on the y-axis here, and on the x-axis is the clinical data. And you see that allowed us to generate a very robust correlation, which allowed us to generate our correlation equations that we can then use to back calculate when we have an unknown compound. And then the next slide is just an example of how we would then use that data, or how you use this data to predict free clinical exposure associated with this 10 millisecond change in QT. So again, in this case, same graph, but you can imagine this is your unknown compound. You run your compound, you generate the concentration, the threshold concentration here. You can then put that back into the equation you generated previously and do the backward calculation and predict the free clinical concentration producing that 10 millisecond change in QT. Now, one of the differences here is it can be an association or there can be a prediction. And prediction requires a little bit more statistics that we performed. So we performed a four-fold cross-validation of the data, which allowed us to show that the assay was actually predictive, and we got some very robust data, information from that. And the assay, the error on the assay is roughly about fourfold, but in reality, when we've looked at some compounds, clinical compounds, we're finding it's more in the range of 2 to 3-fold. What's nice as well for this assay is we do have published data available for an additional 43 reference compounds, and it's all available in that publication as supplemental data, and that results in a total of 66 compounds that are available for anyone to look at and use that data if you want to mine that data in any way. And of course, that includes all the CYP28 compounds as well. So now I'll move on from looking at QT probability to another issue that's in the clinic that is probably less talked about, but is still equally as significant a risk, and that's QRS prolongation, which also can result in arrhythmia. And I think the FDA is starting to get a little bit more involved in looking at this as well more recently. So action potential rise time, as I mentioned, is one of the endpoints we look at, and that is associated with NaV1.5 channel activity. So if you block NaV1.5 activity, that can be associated, or sorry, blockade of NaV1.5 activity can be associated with a QRS prolongation in the clinic. And the stem cell cardiomyocyte, we use the rise time to really, as a surrogate to assess potential clinical QRS risk. So how did we do this? Well, the available clinical QRS data is less defined than for QTc. It's more binary in nature. So it's either reported that there is a QTc response, sorry, there is a QRS response, or there is not a QRS response. So we have clinically defined QRS data. The data we used had to be associated with a preclinical exposure. So we knew which concentration of a compound you were actually seeing this QRS effect at. We then compared that to the stem cell data using the rise time threshold concentration. So similar to the action potential duration at the concentration where you broke that sort of threshold value in your analysis. The analysis was performed using 22 QRS positive compounds and 12 QRS negative compounds. And again, the comparison we did was, this time we had to use an ROC analysis, which is shown here on the right-hand side. But what we compared was the QRS effect, so either binary yes and no, versus the ratio of the stem cell rise time threshold concentration over the free clinical exposure associated with that finding. And what we found was that the AROC value, whether we looked at 30 minutes or 24 hours, we get very robust AROC values showing a very good translation of the data, very high sensitivity and specificity. In fact, the values for most of these were identical between the two time points, and the cut point was a little bit different between the two. But overall, it was a very robust assay at being able to highlight QRS risk. When you start to look at the probability, again, we can't look at predictivity from this assay because the statistics will not allow us to do that, but we can look at probability. And if you look at QRS probability at a clinical exposure equivalent to the rise time threshold. So this, again, this is when in the stem cell assay where it breaks that threshold, if your clinical exposure is at that sort of threshold concentration, the probability of a QRS liability in the clinic is around about 74 to 76%. If you then have if your exposure is much lower than that concentration, and it's actually lower than the cut point for those assays, and that was 33-fold for the 30 minutes or 56-fold for the 24 hours. If you have a margin that's greater than that, then your the probability of QRS liability decreases to nominal values of around about 1%. Now this analysis is kind of difficult because it's kind of assumes you have to understand NaV1.5 prevalence, and here the prevalences we're using are quite high. This is from screening data. It's probably lower than this, so these values probably are lower. But a nice analysis over on the right-hand side here is that Derek actually performed was to look at the effect of the stem cell data on QRS risk post NAV 1.5 patch. So this is a risk of your compound that's associated with looking at the patch data. And if you have a positive or a negative response, you can see that it shifts the probability one way or the other. And so it's again, the stem cell data has some utility in really helping you understand the impact of that data on your risk with regards to QRS. And finally, what I'd like to just touch on is just a case study. And we have a number of case studies. Unfortunately, most of them are confidential, but one of our clients was kind enough to allow us to share this case study. So this was one where a compound, and I think this really highlights the advantage of using stem cell cardiomyocytes. So we have a compound here that has a HERG IC50 around about 20 micromolar, NAV 1.5 in a primary patch assay of around about 100 micromolar, and CAV 1.2 where it's almost 300 micromolar. However, when you look in the clinic, you see both QTc and probably more. The QTc is not unexpected, I suppose, but the QRS would be slightly unexpected. You see a prolongation in both at a free clinical exposure of around about 1 micromolar. And again, given the NAV 1.5 patch data, this seems a little bit, at odds with the patch data. When we threw this into our stem cell cardiomyocyte assay, we predicted a 10 millisecond change in QTc plus a significant risk of QRS at or around 1 micromolar free exposure. Now, the disconnect here is likely due to the fact that when we did further analysis, the NAV 1.5 block had components of both state and use dependency, and we feel that in the stem cell cardiomyocyte, the combination of the increased depolarization that you're seeing because of the HERG block probably had meant that the NAV channel was in a more inactivated state for a longer period of time, which resulted in the compound being more potent in a stem cell model than you'd expected to see in a patch clamp assay. So again, holistic stem cell cardiomyocyte model was able to predict effects that were missed in routine ion channel profiling. So it really has a strong utility. So I won't go through this in detail, but this is just a summary. Again, just the fact that we have a really nice stem cell cardiomyocyte model, and it's valuable in assessing CV risk, both with regards to QTc and QRS. And it also, looking at the traces, it does provide a mechanistic insight as to what the CV effects are driven by. Again, whether it be HERG, NAV, or CAV. And what's nice is you can look both acutely and chronically for compounds. This is one assay where, unlike most patch clamp assays where you're limited by the duration you can incubate the compound with the cells, in this case you can actually extend that. So if you have a slow on rate compound, then this is a really useful assay for those purposes. And I'll stop there in the interest of time, so we have enough time for questions. But any questions after the presentation that are not answered, please feel free to reach out to me. I'll be more than happy to help.
Right. Thanks very much, Steve. That's really great. So yeah, so it's well, thanks for the presentation, Steve, and also Derek. And it's time to talk about go through the questions. Okay. So yeah. Okay. Great. Okay. So yeah, there's one interesting one for you, Derek. Actually, so it's so basically it's about legacy data. So our sponsors currently being held to the new ICH E14S7B Q&A standard. So what if you have, GLP HERG data that was generated using different protocols?
Yeah, I mean, this is a tough one for everyone right now. It's a situation that we've been in before. When ICH E14 was first introduced, we had kind of some of this situation where we had a lag between the introduction of new guidance and everybody's assays and clinical studies catching up. We've submitted a number of waiver requests based on legacy data. I have to say that we don't win them all. We've had some regulators have said, yeah, this HERG's fine, even though it wasn't best practice. We've had others that have kind of been more insistent on us doing a new HERG study. In general, if you have a very large margin, of course, it's easier to argue that even if you did have some deviations from what might be best practice, given that you've got a large margin, it's unlikely that margin would erode. But you also have to have some supporting data with the reference agent. So if you've got in the same format, you've got some moxifloxacin data and some dofetilide data, then that you can use that. But I will say then you've got the other issue that one regulator may say, yeah, you know what, I'll accept your legacy herd, but another one doesn't. So we find ourselves in the situation now of doing repeat hergs. So we have had to repeat hergs.
Okay, brilliant. And actually, yeah, another really interesting one for you, Derek, actually. So what about if you have a compound that's for a life-threatening condition, for example, in the oncology field, and where there are no current therapies, and you get a finding in E14S7B? So what, did it still prohibit approval?
So those are situations where, I mean, this is really telling you, do you or do you not have the risk? And then risk benefit is the second question, and this question is all about risk benefit. And we have seen that there have been a number of compounds that have been approved with QT prolongation, and if they've got QT prolongation, then they presumably also had narrow margins in hERG and in vivo, and those compounds are approved. Obviously, it depends on the indication and some of those considerations. It'll always be a review question, so I don't think you'll get an early answer from the regulators, because they want to see what the entire package looks like in the end and how good your efficacy is, because it's all very well saying it's for a life-threatening indication, but it has to work to make it to really demonstrate the benefit relative to that risk. But yeah, that's a risk-benefit situation. So there's no, you can move forward with some degree of risk.
Okay, great. Right, Steve, I have a question for you, and it's so basically, does the assay that you just presented on, does it actually meet the standards outlined in the updated S7B Q&As for regulatory decision making?
Yeah, when the assay was developed, we just basically went through the various sections of the guidance and tried our best to address every single one of them, and I believe we addressed every single one. As I say, in the routine running of this assay, it doesn't make sense to do compound concentration verification for every study, because you're usually using this a little. This is an assay that I tend to use at an earlier stage in assessment, not right at the very end. However, if you need to use it as part of a package, then at that stage you can do the compound concentration verification quite easily. But that the assay is run to the correct standards. We do have the same data as Derek alluded to, the three reference compounds, moxifloxacin, ondansetron, and dofetilide have been running this assay and have been shown to be in alignment with, again, the FDA's data. So yeah, as a package, it's a very tight package.
Okay, brilliant. And Derek, another interesting one. So maybe people saw some of your answers online, but it was are hERG GLP studies required for antibody therapeutics and other biologics?
So generally, the antibodies, the answer is no. There's no requirement for a standalone QTc study for monoclonal antibodies either. But the jury's kind of out on the other biologics. There's a draft guidance from the clinical pharmacology guidance from FDA on oligonucleotides and on peptides. Both of them would suggest that you have to do some work and treat them essentially as small molecules. You can kind of get a pass if your peptide only had natural amino acids, but my experience is that almost every modern drug peptide has some non-natural amino acid added, so that exception doesn't really help you too much. It is an open topic though, so we will go back now as the ICH-S7B E14 group and develop some Q&As around this, because lots of people are asking it, since ICH-S7B was very much created at a time when most companies' portfolios were dominated by small molecules. But actually, if you look at the portfolios of many large companies now, and some obviously small companies focused on biologics, so small molecules have become far rarer than they were at the time of ICH-S7B finalization.
Right. Okay. Brilliant. And then, yeah, I think this is a question for both of you actually, which is, in terms of an E14S7B HERG study, what positive control? Have you got any thoughts on that?
I mean, I'll let Steve. I mean, the folks at Metrion are obviously doing these. I mean, my experience having done electrophysiology, nobody wants to do the Dufetalide because its kinetics are so slow. It's incredibly tedious. So that's probably taken that one off the board. You've got then moxifloxacin and ondansetron. Most people's experience is ondansetron being so fast that actually it's a pretty reasonable positive control to use, but there's probably nothing essentially wrong with moxifloxacin either.
Yeah, and I think when you look at across the various CROs, I think most CROs are focusing in on ondansetron just because of it's well behaved. It's in that sweet spot. It's not too potent. It's not too weak. Moxie had to go quite high. The Dufetalide's too slow. So yeah, I think ondansetron is probably going to be the sort of reference compounds of choice. But again, I think what's important as well is having all those streets, and there is information that Derek's going to be hopefully releasing sometime soon where, across the CROs, it's important to have all three compounds as at least in your back pocket in case that's required for any, regulatory filing. So I think that's important as well.
And if we come back to that legacy question that we had earlier, that's one of the difficulties because the positive control that was used in most of those was either terfenadine or cisapride. So, and neither of those are included in those three. So that's been one of the kind of awkward points with legacy data.
Okay, brilliant. And Steve, yes, here's an interesting question about translation using iPSC, which is if you've got a compound that's hitting multiple ion channels, will it translate? You've clearly shown that it does for HERG. Will it for other compounds?
Yeah, we have. I mean, it's always so. You're always limited by the clinical data you've got, and using reference compounds is great, but reference compounds and then real-world clinical. I mean, I know those are real-world clinical compounds, but moving forward, it's always interesting to see whether it will continue to translate. For the compounds we've seen so far, yes. With regards to specifically multi-ion channel effects, I can't go into too much detail here, but this will be published sometime soon. But bottom line is, if you have a compound, there's a compound that we had that had some, it was a quite potent HERG blocker, and the exposure was going to be right on top of where the HERG block was going to be. So you'd expect CQT in the clinic, but it also had a little bit of weak calcium activity, a little bit like a verapamil in its profile. That compound wasn't allowed to. The TQT waiver was declined on that one, so it had to go through to a full TQT study. And the stem cell data showed that it wouldn't have any QT prolongation, and it didn't in the TQT study. So it was very nice, validation that. So the assay, we've got data showing that the assay will pick up effects. But we also have data for this assay showing that it will show a negative when you would expect to see a negative because of that multi-ion channel effect. So that's reassuring. Because you certainly don't want to be throwing the baby out with the bathwater, and I think that's where the value of this assay is as well.
Okay, fantastic. So I think we've sort of run out of time now. So thanks for the fantastic discussion. We've got loads more questions, but we'll answer them afterwards by email. So well, thanks to everybody. So thanks Derek, and thanks Steve for your fantastic presentations and the really interesting discussion. It'd be great if we continue it, but obviously everyone's scheduled till 5 o'clock here. So yeah, thanks everybody, and thanks for joining. Okay, right. Goodbye everybody.
Bye.
Bye.